Hello, it's a pleasure to be here today. I'm talking about apple breeding, clonally propagated crops, and their complexity. It is interesting to work with a very diverse crop, one that is a hybrid of two different genetic systems, the scion and the rootstock, and to appreciate how much diversity there is and how much can be harnessed for genetic improvement. So you're going to hear from a bunch of us about genes, technologies, new varieties. But what I'd like to do is discuss preparing yourself for a career in plant breeding by following what has set some of us, many of us apart, and realizing that everything is complex. Even stuff that we thought was very simple, we have learned is incredibly complex. And as a researcher, you need to figure out how you're going to look at the micro and macro views of this complexity. Second is that you really need to become an expert in your field. And that means by always reading new research, old research, forming collaborations, and making use of others' expertise. People are incredibly generous and are willing to help, just have to ask. And by trusting your gut. My advisor for my master's degree always talked about serendipity. And he called it being in the right place at the right time with the right set of eyes. And trusting your gut means that you read or see that citation for a paper 4 times and then say, well, maybe I better read that. There's all sorts of opportunities out there. And sometimes the best failure can seem awful at the time, but lead you to new discoveries. So some fast facts on apple. There's now probably over 60,000 genes that have been discovered. We have genomic sequences for Malus domestica, a haploid accession, double haploid accession. We have 17 chromosomes. We have many areas of genetic duplication. Apple has been around forever and has evolved during those times. We have many transposable elements or jumping genes that can activate and deactivate genes. And then these are best known for color, sports, and architecture. But then you have transcription factors that are also complicating things and methylation. We have 25 to 34 different Malus species, and sadly, only a few of them have really been leveraged. We have tremendous genetic and phenotypic diversity. Apple is wild in that extent. We have self-incompatibility and inbreeding depression that reinforces that heterozygosity. And we're clonally propagated. That's a strength because we can fix something, but it's a challenge because there are many viruses that can be difficult to detect. And once detected, can make commercialization a nightmare. We're also marketed by name, which gives you the ability to really capitalize on development of a brand. So you can see the amazing amount of synteny, the activity of transposable elements, the methylation that occurs in apple. So what this means is if you find something, a QTL on chromosome 2, you better also look at chromosome 7 and chromosome 15 because there are areas of duplication. So I love transposons. Somebody else's Twitter account is I heart transposons, but we have a competition in the lab to find the most in the field. This isn't mine, so I'm in the second running, but a lot of different activation of different colors. This russeted chimera was used by one of my graduate students to be able to determine that russeting enhanced the production of fluoridazine. This is an apple that every fruit is chimeric. Gala is similar to that. Seedling chimera, spur habits also imposed by a chimera, and columnar growth habit, which we feel will have amazing potential for robotics, is also conferred by activity of transposons. But as again, I say, nothing is easy. We always used to think that the genetics of apple color were pretty straightforward. What these diagrams are showing you is 4 different color sports of the same variety. It could be delicious or Fuji. And you can see the tremendous difference between these, what we call simple genetic mutations. There's hundreds of genes that are differentially expressed, and there's transcription factors, and there's activation by UV light, by cold temperature, by stress. And so this is where I was saying, understand that complexity truly is complexity, excuse me. Understanding variation. So we know that there's differences depending on crop load, king dominance, or the fact that the king flower is usually the strongest, as is the fruit, and how you sample, whether you choose a king or a lateral, will influence the type of data that you obtain. So you have to be very careful. What's the age of the tree, the rootstock, the season, the sprays used, the stage of maturity. A failure to account for some of these can ruin your experiments. Developmental stages are also important. Ethylene is a major contributor to apple ripening and also influences other traits. So understand that, become best friends with your physiologist or post-harvest physiologist, pathologist, entomologist. So we know that studies are greatly influenced by the maturity of the fruit that you're testing. And when I first started, I realized that the previous breeders were using very ripe fruit as their indice for quality, whereas the growers are picking at this stage. So in numerous genomic and genetic studies that we've done, I've always stressed that we need to have a stage we can characterize. We can use ethylene, that would be great if we could use a DA meter, because by using overripe fruit, a lot of what you hope to learn or to change, it's already too late. So we breed for quality, of course, but also nutrition. We are fortunate that our program has great latitude to study different things. And I remember my own children's toddler stages, which can be incredibly frustrating, but are also helpful for us because we need to be like them and to never lose that questioning of why does that happen? How does that happen? What is it? And reading the literature will help. It's a key to all our success, and we've leveraged that knowledge to make advances. For example, our nutrition research benefited from a very little published or expanded 1962 study. Climate change breeding will be a challenge that we all have to look at and move forward with. So this is the diversity we see just for leaf morphology. So normal apple leaf looks like the smaller leaf on the stone, but we have some that look like rosemary leaves, and you can see that the leaves are actually growing on the trunk, which we've never seen to that extent before. Leaf lobing can appear in one progeny and have very little expression, but be in the background of other progenies. We were able to amp up the expression of red leaf color, and these make wonderful summer scholars or graduate student studies. For flowers, you saw the initial single blossoms, but this is the kind of variation we can have in terms of color, number of blossoms, vibrancy, a lot of double flowers. And we had a summer scholar that determined that some genotypes that are homozygous for double flowers are in fact lethals. Tree form, again, what's not to love about this? So you have trees that have the columnar habit, are segregating for dwarfs, are shaped like a wheel without pruning, weeping. We want branch angles that are about 90 degrees for greatest strength. And this is just a snapshot of some of the diversity. And then I talk about complexity. Never have 2 guys named Ben in your lab because it makes life very interesting. But Ben Orchesky is someone that looked at Malus prunifolia and looked at something I was really intrigued about, which was pale green lethal. A lot of breeders said, we don't see pale green lethal. I saw in a lot of our progenies, 25% of the population would die due to this lethality. And Ben took it on as a side project and did such a good job with it. It became a NEFA-funded doctoral scholarship for him. And we made the connection that vitamin K was involved in pale green lethality. And I had Googled a colleague that contacted me and found out that he was an expert in phyloquinone. Again, serendipity, perhaps, being prepared, perhaps, but he was able to provide us with the means to assay for vitamin K in very small cotyledons. Ben Archesky offered the other Ben Gutierrez, the leftover Malus prunifolia seeds because it seemed that they were segregating for traits that Ben thought was of interest. So Ben Gutierrez surveyed the USDA apple collection at Geneva. He looked at segregation of fluoridazine, dihydrochalcones, fluoridazine, trilobitine, and sibolden, which are important in glucose metabolism, but also in their role as bitter blockers and sweeteners. He conducted GBS on this progeny. And this progeny is segregating for all sorts of amazing things. He convinced me to plant several more of those progenies, and we'll continue to look at this role. Ben is now the curator of the apple germplasm collection, so this collaboration will be long-lived. We'll switch a little bit because I'm talking a lot about a lot of different things, but what's intriguing about this is, as a breeder, as a geneticist, seeing your varieties in the supermarket is absolutely priceless. Having someone write to tell you that it's their favorite apple is absolutely amazing. And these were two apples that we did under a club system where only certain growers, but in this case, all growers in New York State, had the option to apply. The last three varieties that we have released are open releases so that anyone has the opportunity to enjoy them. So in the future, look for a fully russeted apple. It's one of the most amazing tasting apples that I've had the good fortune to try and to develop. And then this is an example of some of the output from our ornamental breeding, which is columnar and double flowered. That's my technician, Kevin Maloney, who is a wonderful resource to the program. So challenges, we're going to have invasives. Every program is going to have invasives. How devastating they are and what we can do about them will require collaboration with entomologists, but we have brown marmorated stink bug, ambrosia beetle, and spotted lanternfly, which are the latest slew of invasives. Climate change, we know that that's going to be a challenge. I told you about it. In the north, we're battling not only frosts, but hail. So that's why we use hail nets. In the Washington, Oregon area, they're battling drought, as opposed to this recent meme about in New York and our rains, but the drought and the heat are causing sunburn. That sunburn becomes a secondary avenue. So how are we going to breed for something that can have such extreme manifestations? And we are still pursuing this. So future opportunities. We have new flavors, new markets, hard cider, fresh cut, healthy foods, new flavors, textures, nutrients. Some of these will be club varieties and others will be open to the public at large. I want to thank you for your attention and for being the next generation to be following in our footsteps. I have never regretted my career choice. A special thanks to the apple industry, Cornell colleagues, my collaborators, our field staff, Kevin Maloney, and former students and summer scholars. If there's time, I'd be glad to open it up for any questions. I thank you again for your attention and for your interest in plant breeding.